My work uses non-invasive MRI methods to study the brainstem dopamine system in humans. The brainstem is at the top of the spinal cord. The dopamine system, the brainstem in general, are a very old brain system. We share them with lizards. The dopamine system is widely affected in drug addiction and numerous mental health disorders. And as a graduate student, I developed a series of novel MR methods that allow precise and high-resolution measurements from these dopaminergic nuclei in humans. And as a postdoc, I've been working to extend those methods to study both the complexity of the human dopamine system and also drug addiction.
My research is focused on understanding how we transition from casual drug use to more compulsive drug use that's kind of seen in addicted individuals. I use preclinical models, so looking at behavior in rodents primarily, and focus on the neural circuits that are involved in addiction. So what we do is we implant a large number of microwires into areas like the prefrontal cortex, which is involved in guiding cognition and behavior, and watch how that representation changes over the course of the transition to drug addiction.
My research focuses on drug addiction and novel ways to treat drug addiction by reducing relapse. Specifically, my research actually focuses on the therapeutic effects of chronic exercise using a rodent model. The reason we're looking at chronic exercise is because some clinical studies and some preclinical studies using rodents have actually shown that chronic exercise can actually reduce relapse. What none of those studies have shown, however, is the mechanism by which this is happening. So what I'm currently trying to do is figure out and uncover what the molecular mechanism by which exercise elicits its therapeutic benefits. What I'd like to see the field in 30 years, that hopefully in 30 years would have a cure for drug addiction. For example, psychostimulant abuse there's no treatment currently, no FDA-approved treatment for psychostimulant abuse. And there are treatments that are FDA-approved for the other types of abuse, such as heroin or morphine or nicotine, but some of those treatments have pretty significant side effects. So it'd be nice to see if we could improve on some of those treatments. Additionally, it'd be interesting to see if we could also improve some of the behavioral therapies that we have, because focusing too much on the pharmacology, I feel, would just be one side of the problem. And this is a disease, a disorder, if you will, that is multifaceted, and so would require a multitude of tools in order to really truly tackle it.
All of our research kind of speaks to the need to develop new ways of understanding how the brain relates to addiction. My work is focused on preclinical models that could then be translated to human models, where you could do imaging research and look at similar changes in the brain, that could then, hopefully, these brain regions could be targeted with treatments and that could ultimately be used for finding cures or preventative techniques that would help us solve this problem.
I'm really excited to see what the relationship between human neuroimaging studies and animal preclinical studies will be within the next 30 years. I think that the track that human neuroimaging is on right now, with some of the high-res functional imaging that's ongoing, and some of the what people are starting to do with DTI imaging as well, that the progression that the human neuroimaging studies are taking will allow for some really successful parallel studies between animals and humans to take place that I think will have a lot of impact on the study of drug addiction, and hopefully we'll be able to actually learn something from all the neuroscience research that's going on that will result in a successful treatment.
So I think all of us as young researchers can hopefully put all of our different techniques together and use them to come up with some new treatment methods. So for example, I could use my preclinical research to develop some new targets for potential treatments, and then that could be used in humans.
Right, and I have neuroimaging methods that I've developed that look at those, the dopamine regions in people.
And of course, that could tie in as well was actually developing treatments that tie in what you actually found in your preclinical research, and can also be corroborated with what's found in imaging research to actually see how it could treat drug addiction and other related disorders.